All right, so we are Accelero Biostructures located in San Francisco. Our focus is on protein crystallography-driven drug discovery, so just not structure-based drug discovery, but really driving it by experimental protein crystallography. And I'll tell you a little bit about screening molecular compound libraries directly by protein X-ray crystallography that we've been working on for about 6 years or more now. Thanks to Christian for the invitation to participate in this meeting, and it's been a great morning so far. And the previous two talks, really, especially by Sergey, already gave a good introduction about what I'm going to talk about. So I can really focus on a very few key points that I would like to make in the time span.
Debanu, we can see the Apple thing popping up, the message. Maybe you can move it. So better.
Okay.
That was clear. Thank you.
Good now? Okay.
Yes.
Yeah. So, I think many of us are used to high throughput crystallography now, or at least thinking about automation and synchrotrons and rapid data collection, but we often forget a bit of, where this originated. So I'd like to just take a step back, present a couple of slides on a historical perspective of where we are and where we came from. So, in the late 1990s, NIH and Accelero were working on human genome sequencing. And at that time, several highly respected senior crystallographers in the community proposed that people should be working on a structural complement to genome sequencing. And in 1998, about 21 years ago now, Sang-Ho Kim at UC Berkeley, Lawrence Berkeley Labs, he published in PNAS this seminal paper called Structure-Based Assignment of the Biochemical Function of a Hypothetical Protein, a test case for structural genomics, and in which he proposed that we are sequencing, with advanced sequencing, just sequencing genomic space, but how do we understand what the proteins are doing? And the fundamental point was, let us try to develop high throughput methods to solve three-dimensional structures rapidly in a method called structural genomics. And this way we would be able to really assign and understand the structure function relationships of novel proteins. And this might be possible with increasing advances in computer hardware and software associated with structure determination. And this would become more viable. So before that, people were doing one structure for several years, and it was very painful all the way from cloning to structure determination. So this was the year I started my PhD, and I was lucky enough when I finished to be able to go to Sang-Ho Kim's lab, where the Berkeley Structural Genomics Center had already started. It had been 3 years, and I got right into the middle of it, working on automation development all the way from cloning to structure determination. So this was in an umbrella called the Protein Structure Initiative that went through 3 phases over 15 years, in which everything that you see now in academia, in universities, in companies, all the way from high throughput protein expression, purification, crystallization, synchrotron tools, structure determination methods, software development was done to do high throughput crystallography over these 15 years. And so after working a few years there, I spent another 8 years at the Joint Center for Structural Genomics, led by Ian Wilson, headquartered at the Scripps Research Institute, with the crystallography group based at SLAC National Lab, again in Menlo Park here in San Francisco. And during this time, after that, in 2015, 6 years ago, we started Accelero Biostructures as a new entity with all the expertise coming from high throughput crystallography. And so we essentially capitalized on 15 years of expertise and methods development, technology, and operational expertise at the JCSG, which was a multi-consortium institute of multiple institutions, where we had developed a robust gene-to-structure pipeline and driven by efficiency and cost. In all, 1,500 novel protein structures were deposited in the protein data bank during that time, and we published 300 papers on structures and methods development. So with all this background in high throughput crystallography, we decided what's next. How do we make this relevant at Accelero Biostructures and bring this to drug discovery and structure-based drug discovery? How will it enable us to tackle more targets? Can we look at more drug development? Can we produce more drugs? Can we do this in less time and cost? And the obvious answer was fragment-based drug discovery was one of the ways to go, to look at experimental 3D structures generation right from the start without doing anything else. And this was also, I should point out that in the early days of structural genomics, there were companies like Sang-Ho Kim's Plexicon and Aztecs, which still exist, as well as Syrix, SGX. They were also developing, trying to look at high throughput for drug discovery. So this was really building up a lot on top of that. So what is fragment-based drug discovery? In traditional high throughput screening, You have a target and you take a molecular library and you screen it. And usually the molecular library is, of course, not target specific, right? It's a generic library with big molecules. And then you try to optimize the hit for the relevant target. Fragment-based drug discovery, of course, works on selecting small drug-like molecules that are smaller, but more target specific because they bind to specific pockets in your target of interest to be able to generate, with the hope of being able to lead to a more target specific drug at the end. So this is, of course, now widely used to provide novel methods for developing new therapies, in fact, FBDD in general, and it is very efficient for scanning chemical space with a small number of compounds, typically around 3,000 to 4,000 covers your wide variety of chemical space, as opposed to billions of compounds in DNA encoded libraries or millions of compounds being tested in virtual screening, and even hundreds of thousands of compounds in traditional high throughput screening. So with this, this actually makes it suitable for doing by X-ray crystallography. And the advantage there is that Sergey pointed out a lot as well, that often the other biophysical methods for screening, like SPR, thermal shift, NMR, they are often used to pre-screen fragments, but you have no idea how they're binding, where they're binding, what the binding pose is, nothing, right? So you cannot have any actionable evidence on or decision making on chemistry and biology just based on that. So what we have done is when we started this 5 years ago to actually 6 years ago, to develop this platform for crystallography-based fragment screening, which can accelerate by directly providing 3D structures of fragments bound to a target of interest, potentially leading to 5 to 7 times reduction in early drug discovery times, and providing an unprecedented experimental route to high quality, high reliability, and high value results simultaneously, where we can identify and assess fragment binding sites and poses, target ligandability, and differentiation of orthosteric and allosteric sites and binders. So we are now up to, this table shows about 10 or 11 projects that we have gone through, and a number of compounds we test. Typically, we are now all the way up to 300, or in some cases, we have gone to 500. We can now actually go all the way with our proprietary fragment library, proprietary crystallization and crystallography, and this is all in-house and owned by Accelero. So we do not utilize any other public or shared research facilities to do this. We have built up all our entire high-performance computing, in-house crystallization and high-throughput crystallization, imaging, and everything. So we can now go up to 1,000 fragments in a week. So which means that once we have a crystal system set up for high-throughput screening, we can scale up the crystallization, the crystallography, screen the compounds, collect the data, solve the structures, and analyze the results all within a week for about 1,000 fragments at a time. And of course, this is all backed by over 20 years of expertise in developing crystallography and high throughput methods, because there are many, many pitfalls along this whole process that Sergey also pointed out. And you can, if you do not know what you're doing, you will just get junk out of it. So this is very, very problematic. Hit rates, of course, depend a lot on the target size, how big the protein is, what kind of potential binding sites it may have in terms of cavities and so on. But we can, get a pretty good hit rate. These are some of the examples we have had. I'll just quickly go through them. You can see the kind of electron density maps that you can get and see where your molecules are bound. So last minute, I'm going to just take a minute to say how we're translating all this into drug discovery and therapeutics development. So we started a new company which is taking results, which has taken some of these results out for specific DNA damage response targets in oncology to be able to develop therapeutics out of them. So, there are roughly 5,000 new cancer cases per day in the US, of course, many more around the world, and this remains an unmet medical need. But it is also known for many years that there are inherited or sporadic effects in DNA that result from endogenous and exogenous factors, and there are multiple pathways involved in this that eventually lead to cancer. And until about 5 years ago, this was all in the realm of academia until the Nobel Prize in 2015 on DNA repair, after which there are 15, 16 companies working on this. We started working on this problem at Accelero even in the year the Nobel Prize was awarded, before the Nobel Prize. So we had an early lead, but at that time there was hardly any company working on it. And now we've actually spun out a different company to pursue this all together. So in this company, we have built a DNA DDR platform, as we are calling it, DNA damage response drug discovery platform, the center of which is high throughput crystallography, and driven by fragment-based drug discovery. And of course, we can take methods, hits from other methods as well, but then pipe it through this iterative process of structure-guided optimization and then downstream biology. A couple of examples from here. So this is a protein that is target that is highly expressed in rapidly replicating cancer cells in about 20 cancers, and it has a broad applicability in a
Debanu,
bunch
you have
of
1 minute left.
yeah. So we have this is my second slide. I'll be done. So we actually have the first drug-like molecules bound to this target, and an example of this, kind of nice density that you see, and you immediately know where everything is happening, right? This is another target where we have also, again, the first drug-like small molecules bound to the targets. And in both these cases, we have been able to, this is an electron density of a particular hit we got. But in both these cases, we have now been able to move these very quickly from weak hit to functionally active binders, even though just in the first step of chemical modification, we have been able to get some functional activity already, and we are really working on this. So it gives a flavor of how we can rapidly use do drug discovery based on hits taken directly from X-ray crystallography. Thanks. I'll stop there.